हेलो एवरीबॉडी, वेलकम बैक टू माय चैनल। आज हम ह्यूमन रिप्रोडक्शन के सारे डायग्राम्स को डिटेल्स में समझेंगे। लेट्स बिगिन बाय अंडरस्टैंडिंग द मेल रिप्रोडक्टिव सिस्टम। सो हियर वी हैव अ टेस्टिस, द प्राइमरी मेल रिप्रोडक्टिव organ. It produces sperm and the hormone testosterone. The scrotum holds the testis outside the abdominal cavity and maintain a lower temperature, जो spermatogenesis के लिए essential होता है. The coil structure is the epididymis. Yahi sperms get mature and are temporarily stored. From epididymis, sperm enters the vas deferens, a muscular tube that transports them upward. The seminal vesicle adds a fructose-rich secretion providing energy to sperms. The vas deferens joins to the duct of the seminal vesicle to form the ejaculatory duct. The ejaculatory duct passes through the prostate gland, which adds an alkaline secretion to increase sperm motility. These are the bulbourethral gland. They release a clear lubricating fluid that neutralizes urethral acidity. And the urethra is a common passage for urine and semen, but never at the same time. The penis is a copulatory organ. The glans penis is highly sensitive and is covered with a foreskin. The next is the diagrammatic view of male reproductive system, where a part of testis is open to show inner details. Each testis is divided into multiple testicular lobules. Inside every lobule, we find highly coiled seminiferous tubules. यही actual site है sperm formation का, that is also known as spermatogenesis. From the seminiferous tubules, sperms are collected into a network of tubules called the retrotestis. Retrotestis acts as a collecting channel, collecting sperms from different tubules. From the retrotestis, sperm passes into small ducts known as vasa efferentia. These ducts help in transporting sperm out of the testis. The vasa efferentia opens into epididymis. This is long coiled structure and is divided into head, body and tail. Within the epididymis, sperm undergoes maturation and gain motility. यही sperm temporarily store होते हैं before further transport. From the tail of epididymis, the duct continues as vas deferens, marking the end of the testicular phase of sperm transport. So this diagram specifically explains the internal pathway of sperms from seminiferous tubules to epididymis. Once this sequence is clear, diagrams related to male reproduction becomes very easy to understand. Now let's discuss about the microscopic structure of seminiferous tubule. These cells present outside the seminiferous tubules are the interstitial cells, also known as Leydig cells. Their main function is to secrete the male hormone testosterone, which regulates spermatogenesis. Near the basement membrane of the seminiferous tubule, we find spermatogonia. These are the male germ cells. They divide mitotically to maintain the sperm-producing cell population. As spermatogonia moves towards the lumen, they undergo differentiation and maturation. This gradual transformation is the core of spermatogenesis. And these long column-like cells are Sertoli cells. They extend from the basement membrane to the lumen. Sertoli cells provide nourishment, protection and structural support to developing sperm. इसीलिए इन्हें nurse cells भी कहा जाता है. Near the lumen, we can see fully formed spermatozoa. These are the mature male gametes released into the lumen of the seminiferous tubule. So this diagram explains how sperm are formed at the microscopic level from spermatogonia to spermatozoa with the support of Sertoli cells and hormonal control by the Leydig cells. So once this diagram is clear, spermatogenesis becomes very easy to visualize. The next diagram is of the sectional view of female pelvis showing the reproductive system. The central muscular organ is the uterus. It is the site where implantation occurs and where the embryo develops during pregnancy. The lower narrow part of the uterus is called the cervix. It connects the uterus with the vagina and regulates the passage between them. This muscular tube is the vagina. It receives the male gametes during copulation and also acts as the birth canal. This external opening is the vaginal orifice, which opens into the outside of the body. These outer folds of skin are called the labium majora. They protect the external genital structure. Just inside are the labium minora, smaller folds that surrounds the vaginal opening. This small sensitive structure is the clitoris. It is rich in nerve endings and is involved in sexual stimulation. The short tube is urethra, which carries urine from the urinary bladder to the outside. Located anteriorly is the urinary bladder, part of the excretory system, shown here for positional understanding. This bony joint is the pubic symphysis, which provides support to the pelvic region. Posterior to the vagina lies the rectum, part of the digestive system. And the terminal opening of the digestive tract is the anus, shown here to understand the anatomical relations. And understanding this spatial arrangement makes topics like menstrual cycle, fertilization, pregnancy, and parturition much easier to visualize. So the next is the frontal view of female reproductive system, showing uterus, oviducts, and the ovaries. These oval structure on both sides are the ovaries. Ovaries are the primary female reproductive organs. They produce female gametes, ova, and also secrete the hormone estrogen and progesterone. At the free end of the oviducts, we see the finger-like projections called fimbriae. Their role is to collect the ovum released from the ovary. This funnel-shaped part of the oviduct is infundibulum. It receives ovum after ovulation. The wider middle portion of the oviduct is the ampulla. This is the most common site of fertilization. The narrow part that connects the oviduct to the uterus is the isthmus. It helps in transporting the fertilized egg towards the uterus. And the spear-shaped central organ is the uterus. It is also called the womb. The uterus is also the site of implantation, and it supports the development of the embryo and the fetus during pregnancy. The inner lining of the uterus is the endometrium. It undergoes cyclic changes during the menstrual cycle. The thick muscular layer is the myometrium. It plays an important role during childbirth by producing strong contractions, and the outermost layer is perimetrium. The lower narrow part of the uterus is the cervix. It connects the uterus within the vagina. This canal present within the cervix is called the cervical canal. It forms a part of birth canal. The next part is vagina, and this part is also known as uterine cavity. The next is the diagrammatic sectional view of mammary gland. The large glandular units are the mammary lobes. Each mammary gland consists of several lobes. Each mammary lobe is further divided into smaller units called mammary lobules, and these lobules contain milk-secreting cells called alveoli. Within the lobule, we see secondary mammary tubules. These tubules collect milk produced by the glandular cells, and several secondary mammary tubules join together to form the mammary duct. Near the nipple, each mammary duct slightly enlarges to form an ampulla. Ampulla acts as a temporary storage site for milk, and from the ampulla, milk passes into lactiferous ducts, which opens on the surface of the nipple. This projecting structure is the nipple. Milk is released from the nipple during lactation, and the pigmented area surrounding the nipple is called the areola. It contains sebaceous glands that help in lubrication. This yellow region represents the fat tissue. It provides shape and support to the breast. And behind the mammary gland lies the pectoralis major muscle. It provides a muscular base, and the ribs and the muscle between the ribs are shown here for anatomical support and position. So this diagram explains the structural organization of mammary gland, from milk secreting lobules to milk releasing lactiferous ducts. Let's move towards the next diagram, which shows the sectional view of seminiferous tubule. The cell present near the basement membrane is spermatogonium. They are diploid germ cells and divide by mitosis to maintain the germ cell population. Some spermatogonia differentiate and form primary spermatocyte. Primary spermatocytes are diploid and undergo meiosis I. After meiosis I, each primary spermatocytes form two secondary spermatocytes. These cells are haploid and they quickly enter meiosis II. Meiosis II results in the formation of spermatids. Spermatids are haploid, round cells and are non-motile. These elongated, flagellated cells are spermatozoa. The mature male gametes, they are released into the lumen of seminiferous tubule. These large supporting cells is the Sertoli cells. It extends from the basement membrane to the lumen. So this diagram shows spermatogenesis in a clear vertical sequence, from spermatogonium to primary spermatocyte to secondary spermatocyte to spermatid and then spermatozoa. If you remember this flow, spermatogenesis becomes one of the easiest scoring topics in NCERT. The next diagram is the structure of human sperm. The entire sperm is covered by plasma membrane, which encloses all internal components. This region is the head of the sperm. At the anterior end of the head is the acrosome. It contains hydrolytic enzymes that help the sperm penetrate the ovum during fertilization. Below the acrosome lies the nucleus, which contains the haploid chromosomal material. Just below the head is the neck. It connects the head to the middle piece and contains centrioles. And this region is the middle piece. The middle piece is packed with mitochondria. These mitochondria provides energy for sperm movement, इसलिए sperm continuous swim कर पाते हैं. The long posterior part is the tail. It helps in locomotion, allowing the sperm to move forward in female reproductive tract. So by the help of this diagram, we understand how the structure of sperm is perfectly adapted to its function. The head is for fertilization, the middle piece is for energy, and the tail is for movement. So here is the sectional view of ovary. The entire structure is the ovary, the primary female reproductive organ. It produces ova and secretes female hormones. These structures are blood vessels which supply oxygen, nutrients, and hormones to the ovarian tissues. These small developing structures are primary follicle. Each primary follicle contains a primary oocyte surrounded by follicular cells. As the follicle grows, it develops into a tertiary follicle. At this stage, the follicle increases in size and complexity. This fully mature follicle is the Graafian follicle. It contains a mature secondary oocyte. During ovulation, the Graafian follicle ruptures and releases the ovum from the ovary. After the ovulation, the remaining part of the Graafian follicle transforms into a corpus luteum. Corpus luteum secretes progesterone, which is essential for maintenance of pregnancy. And this diagram shows the sequence of follicular development in the ovary from primary follicle to corpus luteum. If this flow is clear, ovarian cycle and hormonal control becomes very easy to understand. So here is a comparison between spermatogenesis and oogenesis. At first, let's understand about spermatogenesis. These diploid cells are spermatogonia. They undergo mitosis and differentiate to form primary spermatocytes. Primary spermatocytes are diploid cells with 46 chromosomes. They enter the first meiotic division. After meiosis 1, each primary spermatocytes form two secondary spermatocytes which are haploid and contains 23 chromosomes. Secondary spermatocytes undergo second meiotic division to form spermatids. Spermatids then undergo differentiation called spermiogenesis and form spermatozoa. Spermatogenesis starts at puberty and continues throughout adult life. Now let's understand about oogenesis. These diploid cells are oogonia. They multiply by mitosis during fetal life. Oogonia differentiate into primary oocytes which enter meiosis 1 but remain arrested till puberty. Meiosis 1 remain incomplete from fetal life to puberty. At puberty, during each menstrual cycle, meiosis 1 is completed, forming a secondary oocyte and a first polar body. The secondary oocyte enter meiosis 2, which is completed only after fertilization, resulting in the formation of an ovum and a second polar body. Let's understand about the various events during a menstrual cycle. This cycle begins with menstruation from day 1 to day 5. Here the endometrium sheds, resulting in menstrual flow. This shedding of the uterine lining occurs due to the fall in estrogen and progesterone levels. After menstruation, the uterine lining starts regenerating and thickening. This phase is called the follicular phase or proliferative phase. During this time, ovarian follicle starts developing. The pituitary hormone FSH stimulates the growth of the ovarian follicle. Developing follicle secretes estrogen, which causes proliferation of the endometrium. A sharp rise called the LH surge occurs around day 14. This LH surge triggers ovulation, the release of a mature ovum from the ovary. This is the most important hormonal event of the cycle. After ovulation, the ruptured follicle transforms into corpus luteum. The corpus luteum secretes progesterone, which maintains and thickens the endometrium, and this prepares the uterus for possible implantation. And if fertilization doesn't take place, the corpus luteum degenerates. As progesterone and estrogen level falls, the endometrium breaks down. This leads to menstruation and a new cycle begins. This diagram beautifully links ovarian events, uterine changes, and hormonal function into one coordinated cycle. In this diagram, ovum is surrounded by few sperms. The central spherical cell is the ovum, the female gamete. Surrounding the ovum is a thick non-cellular layer called the zona pellucida. It plays a crucial role in species-specific sperm binding. The narrow region is the perivitelline space. It lies between the plasma membrane of the ovum and the zona pellucida. The cells surrounding the ovum are called the cells of corona radiata. They are follicular cells that provide protection and nourishment to the ovum. These elongated structures are the sperms, the male gametes moving towards the ovum. When a sperm comes in contact with the zona pellucida layer, it triggers acrosomal reactions. During this reaction, enzymes released from the acrosome help the sperm to penetrate the corona radiata and zona pellucida. Remember, only one sperm is allowed to fuse the ovum. After sperm entry, the zona pellucida undergoes changes to prevent polyspermy. Once you understand this diagram, the entire fertilization process becomes very easy to understand. So the next diagram explains the transport of ovum, fertilization and passage of growing embryo through fallopian tube. The first figure shows the secondary oocyte surrounded by zona pellucida and corona radiata. Here, multiple sperm approaches the ovum, but only one sperm successfully enters. After sperm entry, fertilization is completed and the zygote is formed. The zygote is diploid. The zygote now starts cleavage, dividing mitotically into blastomeres. Continuous cleavage results in a solid bolus of cells called morula. Fertilization occurs usually in the ampullary isthmic junction of the oviduct. As cleavage continues, the dividing embryo moves towards the uterus. The morula enters the uterus and transforms into a blastocyst. This shows a compact group of cells, the morula. This hollow structure is the blastocyst. The outer layer is trophoblast, which helps in implantation, and the inner cell mass develops into embryo proper. The blastocyst attached to the endometrium of the uterus. This process is called the implantation, and implantation usually occurs about 6 to 7 days after fertilization. Now let's move towards the next diagram, which shows the developing embryo in uterus showing placenta. This outer thick muscular structure is the uterine wall. It provides support and protection to the developing embryo. This red, highly vascular lining is the endometrium. It is the site where the embryo gets implanted. This structure inside the uterine cavity is the developing embryo. It grows and differentiates into fetus during pregnancy. The embryo is surrounded by the amnion, forming the amniotic sac. The fluid inside is amniotic fluid, which protects the fetus from shocks. एक natural cushion की तरह. The disc-shaped structure is the placenta. Placenta acts as a temporary organ connecting the mother and the fetus. These finger-like projections are placental villi. They increase the surface area for exchange of materials. Maternal blood flows around this villi, allowing exchange of oxygen, nutrients, and waste products. This tube-like structure is the umbilical cord. It connects the fetus to the placenta. The umbilical cord contains blood vessels that transport oxygen and nutrients to the fetus and carry waste back to the mother. Remember, maternal blood and fetal blood do not mix directly. Exchange occurs through the placental membrane. So this diagram explains how the developing embryo is nourished and protected inside the uterus. The placenta acts as the lifeline between the mother and fetus, ensuring growth and survival throughout the pregnancy. With this, all NCERT diagrams are completed. Revise these diagrams again and again, because in NCERT, diagrams don't support concepts. They are the concepts. So see you in the next chapter. Don't forget to hit that subscribe button. Thanks for watching.